fast bacteria in detail. Before that, we have another types also. So next one is the flagella stain. So flagella stain is nothing. It is created or it is developed to see the bacterial flagella. So every bacteria have cell wall or without cell wall, without flagella in its structure. So if the bacteria has flagella, it is having the motile property. So motile is nothing but the movement. So if the bacteria have flagella, it can able to move from one place to another. And if the bacteria lacks flagella, it can't able to move or motile. So it just stays in a very only one place. So in this flagella stain, we need to add silver nitrate, and silver nitrate makes the flagella appear larger than it is actual, and it can be used to determine the arrangement of flagella for the identification. So coming to the arrangement, so flagella are of different types in the bacterial cell wall. So you could see the microscopic images of 3 ABC. So ABC will vary with one another. So these are the flagella types you could see. You can mention it as A-trichus. So A-trichus in the sense the flagella does not having, the bacteria does not having flagella. And coming to the monotrichus, mono means one. So it is having only one flagella at one end. And amphitrichus, it is having only one flagella at both the ends. And lopotrichus, it has more than one flagella in one part of the bacterial cell. And amphilopotrichus, you could see more than one flagella in both the ends of the bacteria. And coming to the peritrichus, you can see the flagella all over the bacterial cell. Okay, so these are the arrangement of flagella for the identification whether it is atrichus, monotrichus, amphitrichus, lopotrichus or peritrichus. So you could see these flagellar arrangement in the flagella staining. Yes, as I said before, today we are going to see the full acid fast staining. So what are all the components used for the staining, what are all the principle, what is the procedure, what is the upcoming observation and interpretation you all see in this class. So before entering into this staining, we need to know acid fast bacteria. So as I said before, this acid fast staining is mainly used to differentiate the acid fast bacterium and non-acid fast bacterium. Okay, so coming to the acid fast bacteria. So this bacteria has higher amount of glycolipids which is mycolic acid and lipoarabinomannan fatty acids. So a very good example of acid fast bacteria is Mycobacterium tuberculosis. So you could note the mycolic acid in Mycobacterium species. Okay, so wherever the Mycobacterium is there, that bacterium has mycolic acid in their cell wall. Okay, so this mycolic acid plays a major role in the acid fast bacteria, and the non-acid fast bacteria which lacks the mycolic acid in their cell wall. So apart from the mycolic acid, there are several layers which is involved in the acid fast bacteria. So that I mentioned as layer 1, layer 2, layer 3 and layer 4 and 5. So layer 1 is nothing but the thin layer of peptidoglycan layer. So only 1 or 2 layer of peptidoglycan layer is present in the acid fast bacteria. And layer 2, it is having the arabinogalactan layer. So you could see both the combinations of arabinose and galactose. So arabinose and galactose is all a form of sugar molecules. And layer 3, it is mycolic acid, and it contains lipoarabinomannan and phosphatidylinositol monocytes, that is PIM. And layer 4 surface proteins, and layer 5 is periplasm region. So this is the structure of the acid fast bacteria. So you could see the components of porins, surface proteins, acyl lipids, mycolic acid. So mycolic acid is a very thick layer in this acid fast bacteria, and you could see the arabinogalactan layer, and you could see the lipoarabinomannan layer, and you could see the very thin layer of peptidoglycan layer, and PIM is nothing but phosphatidylinositol mannositol, and later on the cytoplasmic membrane. So this is the overall structure of the acid fast bacterium, where the components are stacking in the form of layers, right? Yes. So as I said before, these are the examples of acid fast bacteria and non-acid fast bacteria. So acid fast bacteria examples, as I said, Mycobacterium is the more common species for acid fast bacteria. So Mycobacterium tuberculosis, Mycobacterium leprae, and Mycobacterium bovis. So these are the major examples of acid fast bacteria. So Mycobacterium tuberculosis is the causative agent for causing tuberculosis in humans, and Mycobacterium leprae is the causative agent for leprosy in humans. And coming to the examples of non-acid fast, E. coli, Escherichia coli and Staphylococcus aureus plays a major role in the examples of non-acid fast bacteria because it lacks the mycolic acid in its structure, whereas acid fast bacteria having mycolic acid, non-acid fast does not have any mycolic acid in their structure. Yes. So next, coming to the staining. So you need, you have I have explained the acid fast bacteria, what is the difference and what are the components present in that acid fast bacteria. And coming to the acid fast staining, so coming to the discovery, this was originally discovered by the scientist Paul Eldridge in 1882, and later it was slightly modified by the scientists Zeal and Nielsen in 1883. So that this acid fast is also called as Zeal-Nielsen staining, right? So Zeal-Nielsen staining, on behalf of the scientists who discovered this staining, they named as Zeal-Nielsen staining. So both are same. Acid fast staining are also the same, and Zeal-Nielsen staining is also the same. So as I said before, it is a main form of differential staining which is used to distinguish between the acid fast bacteria and non-acid fast bacteria.